Hello everyone, welcome to my channel. My channel name is Biology Lover. Today's topic is molecular means one shot in molecular basis of inheritance means we can uh, discuss about the RNA and the DNA in detail. Okay, so our first topic in this chapter is nucleic acid. We all know that the examples of nucleic acid is DNA and RNA. DNA is the main genetic material that we discussed in detail after some time. Okay, so nucleic acid is the polymers of nucleotides. Okay, just we uh, connect the terms that nucleotide is equal to nucleoside plus phosphate. Okay, nucleoside is equal to sugar plus nitrogen base. Okay, sugar equals to ribose for RNA and deoxyribose for DNA. Okay, nitrogen base are of two types, purine and pyrimidines. Okay, purine also have two types, adenine, guanine. Pyrimidine also have two types in RNA and two types in DNA. For purine means for pyrimidine, there are thymine and cytosine for DNA and cytosine and uracil for RNA. Okay. So next topic is deoxyribonucleic acid or DNA. Okay, this is identified by Frederick Mescher in 1869. This point is most important for NEET examination. Okay, DNA is acidic in nature. This is also the important topic or the important point. Okay, then next point is Wilkins and Franklin studied DNA with the help of X-ray crystallography. They are studied about the molecular genetics by the help of X-ray crystallography. Okay, by this the topic this the crystallography Watson and Crick used, and uh, they produced that the DNA has double helix model. Okay, so adenine, thymine, guanine and cytosine are the four basic nitrogen bond. Okay, so adenine and thymine are bonded with double hydrogen bond. Okay, and guanine and cytosine bonded with triple hydrogen bond. These are the hydrogen bonds that the nitrogen bases are connected to each other. This is Kargaf's rule that purine is equals to pyrimidine, means adenine plus guanine is equals to thymine plus cytosine. Okay, so this is the actual structure, means the innermost structure of DNA. Okay, so these are the these are the sugars and these are the phosphates. These are nitrogen base. Okay, so we also known as that DNA is phosphodiester bond. Phosphodiester means two ester. Esterification is uh, means done in that bonded. Okay, so in previous topic we studied that adenine is bonded double hydrogen bond with thymine and guanine is triple hydrogen bonded with cytosine. These are the these two are diester bond. Okay, so this part is called the backbone of the DNA, means backbone of the this nitrogen bond. These are the these only carry the genetical material, okay, from generation to generation. But the actual structure of DNA is that these are anti-parallel to each other. Means if this pole is 5 prime, but this pole is 3 prime, means anti-parallel. These are not same in uh, one pole to another pole. Okay, so these are nitrogen base. Okay, so the gapping or the distance between the two nitrogen base is 3.4 angstrom. Okay, and in 1 ton, means from this to this is the 1 ton. This the uh, distance is 34 angstrom, means 10 base pair is situated in that 1 ton, means in upper, means in this distance to this distance. Okay, but the distance from this to this, means one end to other end of the nitrogen base is 20 angstrom. Okay, so the question is arised that this long strand or the long chain of DNA, how can it convert into chromosome like a small? Okay, so for this we produce that DNA packaging. Okay, so DNA packaging is our next topic. DNA packaging is like that. The pearl, the means our moms or our sisters wear a pearl necklace like this. The histone proteins are attached with the DNA strand like a pearl necklace. Okay, DNA is tightly packed with histone proteins. Like histone proteins are of 5 types: H1, H2A, H2B, H3 and H4. These are the 5 types of histone proteins. So that long chain of polynucleotide, means the DNA uh, is made up of nucleotide. It is not a single nucleotide. It made up of polymer. Okay, so polynucleotide is take a small place to set up and form a chromosome. Okay, so histone proteins are of 5 types. H1 histone protein is we call as linker protein, means the uh, DNA, the strand of DNA is linked with the histone 1 protein with the first time. Okay, so the histone proteins are arranged in a cube shape. Okay, so histone H1 histone protein is called as linker histone protein, means it has the main role in the packaging, but the side proteins are H2A, H2B, H3 and H4 are arranged in a cube shape. So when we these 4 are into 2, means in a 3D shaped cube shape, then we call as histone octamer, means octa 8 histone protein are produced by the 4 histone protein. Okay, So how can we come from DNA to chromosome is H1 is called linker histone protein, then histone octamer is we said that the cube shape of these four histones, then 200 base pair of DNA means in one histone protein there is 200 base pair of the DNA. Okay, so these all are called nucleosome. Okay, then the sixth nucleosome is formed as a solenoid, means a solenoid form of six nucleosome. Then many solenoid is termed as chromatid, means a chromatid means a single chromosome, single half chromosome. Then two chromatid is from a complete chromosome. This is the pathway that we form DNA to chromosome. Okay. So next topic is transforming principle. We at the first we uh, studied that Frederick Mescher and another scientist is Frederick Griffith is studied about the DNA, that is the transforming principle. It studied about the Streptococcus pneumoniae. The bacteria is Streptococcus pneumoniae. So Streptococcus pneumoniae have two colonies. One is smooth colony, other is rough colony. Smooth colony have capsulated, then it have mucus coated, then it have virulent. Virulent means it easily infect the mice. It studied about on the mice. Okay, it easily infect the mice. But in rough colony, it is non-capsulated. It has no mucus coated, and it is non-virulent. It can't easily um, it can't easily attack in the mice. Okay, when we start our test examination, that mice is died when the virulent is attacked. Then if the virulent is suppressed by heat killed, means the smooth colony is heat killed, then the mice is alive. Okay, but in rough colony case, the mice is already, it has no external force, it is already alive. Okay. Okay, so this is exceptional. Okay, S strand means rough strand, S strand means R strand means rough strand, S strand means smooth strand. We studied about the S strand is non-virulent, S R strand is non-virulent, S strand is virulent. Then if we heat killed that the S strand, it can't affect the mice. Okay, but when these two are not attacked the mice, then why the mice is dead? This is the answer. This is the question arised, but the answer is when the virus of S strand is suppressed, then the virus transformed in the R strand for the for the presentation or their attendance to they are kill the mice. Then the actual solution is the mice is dead for this transforming principle. Okay, so the next topic is biochemical characterization of transforming principle. Okay, so in this in this transforming principle there is three scientists are mm, arranged is Avery, McLeod and McCarthy. That's Avery, McLeod and McCarthy. These are the three scientists who actually studied the transforming substance of DNA. Okay, so they tested with the digestive enzyme, means protein is digested or protein is transformed or the protein is suppressed by its enzyme is protease enzyme. Then RNA is suppressed by the enzyme is RNase. Then DNA is suppressed by the enzyme is DNase. When these three are when these three enzymes are added in three uh, these three materials, then we see that the protease is digested the protein and is transformed. Then RNA is digested the RNA and it also be transformed, uh, but it not suppressed by the RNAs. Okay, but the DNA is not transformed in that digestive enzyme. So actually they called means they examined and they called as DNA the transforming material. Okay, so our next topic is experiment of Hershey and Chase. This is the most important topic in point of view of NEET. Here, the uh, I write this from the NCRT. Okay, so Hershey and Chase are the two scientists who studied with the bacteriophage which is infected into E. coli. Okay, you all know the full form of E. coli is Escherichia coli. Okay, so when the E. coli is affected with bacteriophage, that radioactive sulfur and phosphate are used. Sulfur is for DNA and phosphate is for protein. Sorry, I am sorry. DNA is for uh, phosphate and sulfur is for protein. Okay, so these two scientists are worked this experiment into three steps: infection, blending and centrifugation. Okay, that after this experiment they conclude that DNA contains phosphorus and protein contains sulfur. That unequivocal proof that DNA is the genetic material that E. coli is not transformed into sulfur. It can transform into phosphorus. For that DNA is contained into the phosphorus. Okay. So our next topic is genetic material. We can comparison between RNA and DNA. Okay, means ribonucleic acid and deoxyribonucleic acid. Okay, so what are the properties are present in a true genetic material? Are they show their own replication? Here DNA and RNA both are shown their own replication. Okay, chemically and structurally stable. So here DNA is chemically and structurally stable. For this RNA is not very stable because in 2' strand of RNA this OH bond is present. For this thymine is more stable than uracil. Uracil is present in RNA and thymine is present in DNA. Okay, they changes mutation means they if they change their mutation rapidly, they can't be a true genetic material. So in this case, RNA can change their mutation rapidly. Okay, comparison to DNA. For this, DNA is the best. Okay, and they able to express itself. RNA is able to express itself, but DNA is stronger than the RNA. So at the end, DNA is the true, and the DNA is the main genetically material. Okay, so next topic is lac operon. Okay, so lac operon is the this is the receptor, this is the promoter. Okay, and these are the transcription material. Okay, when repressor mRNA is expressed itself with bond with the O, means O. Okay, it has no anyone to uh, attack. Okay, so that's why it can easily binds with this, then no transcription is occurred. Okay, if here repressor is attacked, or it can't go and bind with this, then transcription is occur, but in this case no transcription occur. Okay, so the next is, okay, in next case, if the repressor mRNA means messenger RNA if not able to bind, means if inducer is suppress the repressor, then the mRNA is not able to bind with O, and it can occur the transcription process. This is beta glycosidic linkage, this is the primase. Okay, and this is the transcytolase. Okay, means the transforming occur. Okay, transcription is occur if the inducer is attacked with the repressor. Okay, so next topic is genetic code. We all know that nitrogen base are of 4 types: adenine, guanine, thymine and cytosine. There are 20 amino acids are present in the 4 nitrogen base. Okay, so if we take 4 to the power 1, it will be 4, then it can't overcome the 20. If we take 4 to the power 2, it comes 16, means it can't overcome the 20. If we take 4 to the power 3, it came 64, means in this case it can overcome the 20 amino acid. So we can call that the codon, means the genetic codon is in triplet in nature, means it is third part of the 4 codon. Okay, total codon is 64. Coding agent means coding the bases are coding are 61 coding, and stop codon are 3. Okay, So stop codons are UAA, UGA, UAG. Means the shortcut in Hindi is Ullu Aya Aya, Ullu Gaya Aya, and Ullu Aya Gaya. This is the means tip to remember that the stop codons are this type. Okay, and starting codon is AUG. AUG is the starting codon, and it is code for the methionine. Okay, in genetic code read in centrifuge fission means no it can read in randomly, not not by that it not by for it is randomly, no comma, no full stop. Okay, it is nearly universal means for all uh, properties it can code. Okay. Code is degenerated means one codon can code only one amino acid. Okay, expression is one codon is code only one amino acid. The scientists are related with genetic code are Gamau, Nenberg, and Kuarna. Okay, so next topic is central dogma. It is the most important topic point of view of NEET. Central dogma is the key point of the molecular genetics. Okay, so central dogma, when we transformed to DNA to RNA, we can say that this is transcription. When we transform RNA to protein, we said that translation. When we reversely transformed into DNA means RNA to DNA, this is reverse transcription. And when DNA is formed their own copy means own Xerox, at this time we can call as replication. Okay, so how transcription is means process. Okay, so for transcription we can take RNA polymerase. For prokaryotes there is one type of RNA polymer which can transcript the three types of RNA: rRNA, mRNA, and tRNA. But for eukaryotes there are three types of RNA: RNA polymerase 1, RNA polymerase 2, and RNA polymerase 3. It codes for rRNA, it codes for mRNA, and hnRNA. It codes for tRNA. Okay, this is the coding agent of the transcription process. Okay, this site we can say that promoter site. This side we can say that terminator site. This is a template strand for mRNA. This is a coding strand means in this strand it can code the RNA. Okay, so it has three phases in transcription process is initiation phase, elongation phase, and termination phase. In initiation phase, RNA polymer is added with sigma factor. Sigma factor is most important in NEET. It can ask you that what factor is used for the code in initiation phase of transcription process. Okay, elongation phase is the RNA polymerase with the rho factor, it also important. And termination phase is RNA polymerase with sigma factor only, not a rho factor. That's why it can cut at the rho factor. Okay. So above the function for prokaryotes, the integer and exon for hnRNA. Okay, that two means this is one for this and for this one for this one, means the other RNAs are present in a single strand. We can stitching this in the continuing strand for 5 prime to 3 prime end. Okay, but the actual is mRNA, which is copying mgPPP, means this is the code for mRNA. This we call tailing, means and adelite residues. Okay, this is 5 prime end to 3 prime end. The above function is for eukaryotes. This is for eukaryotes. This is for eukaryotes. Okay, so if you our last topic in this chapter is DNA fingerprinting. So DNA fingerprinting is our last topic. It has bulk DNA, it has satellite DNA in two types. Bulk DNA can code 99.9% DNA, but the difference of DNA is for satellite. It is non coding. It has 0.1. Okay, it has mini or it has macro. Mini is VNTR, uh, macro is STR. It has 10 to 60 base pair. It has 1 to 8 base pair, but in large size it has 10 to 60, but in small size. Okay, I hope you will enjoy this lecture, or you will gain your knowledge by seeing this lecture. Okay, upcoming lectures are come soon. Okay, I will discuss about the important question of NEET for the for the basis of this chapter. Okay, thank you.